Hello students, welcome to my channel Somalins Bio World. Today I am going to discuss about the structure of and function of nephron. In the previous video, I have already discussed about the internal structure of kidney, and I have already taught that the structural and functional unit of kidney is the nephron. So today we will discuss about the structure and function of nephron. So you can see this is the internal structure of kidney, and these are the medullary pyramids, and this is the cortical part, and this is the medulla part. And here the two blood vessels, they are renal artery and renal vein, they enter into the kidney, and this is the ureter which pass the urine from kidney to the urinary bladder. So this part, the entry, or this is a notch-like structure. This is a notch-like structure where the renal artery, renal vein and ureter, they connect to the each kidney, and this part is known as the hilum. Okay, and I have drawn here one nephron. This is the nephron, and there is about 1 million of neuron present in each kidney. So after the formation of urine in this nephron, then this is the collecting duct. Through this collecting duct, the urine, it comes to this part. This is the renal pelvis, and from this renal pelvis, the urine, it comes out through the ureter into the urinary bladder, and from urinary bladder it finally passes out through the urethra. So come and we will discuss about the structure of nephron. The structure of nephron, it consists of three parts. They are the Malpighian body, the nephric tubule and a collecting tubule. So first come to the Malpighian body. The Malpighian body, it consists of the Bowman's capsule and the glomerulus. So this cup-like structure is the Bowman's capsule, and the blood capillaries, or the network of capillaries, which is in red color, this is the glomerulus. And both these glomerulus and Bowman's capsule, they form the Malpighian body or the renal corpuscle. And the work of both these glomerulus and Bowman's capsule is to filter the blood. So the glomerulus, it has two ends, that is the afferent arteriole and efferent arteriole. When the blood from heart through the renal artery, it comes to the kidney, then the renal artery, again it divided into small arterioles. Again the small arterioles, they again branch and they form the afferent arteriole. And so through the renal artery, the blood from heart, it comes to the nephron for its filtration. And again after the filtration process, the rest of the substance, it again pass out from the glomerulus through efferent arteriole, and again this efferent arteriole, it makes a network around the renal tubule, and again it continue as the renal vein, and through this renal vein again the filtered blood, it go to the heart again. So this is the complete circuit of the filtration of blood. So I talked about the first part, that is the Malpighian body or the renal corpuscle, and it composed of the glomerulus and the Bowman's capsule. And after this Malpighian body, the next part is the nephric tubule, and this nephric tubule has three parts, that is proximal convoluted tubule, loop of Henle, and distal convoluted tubule. So from the neck of the Bowman's capsule up to this part is the proximal convoluted tubule, and this U-shaped loop is the loop of Henle, and the loop of Henle, it continue as the DCT or the distal convoluted tubule. Okay, and here I have drawn a dotted like line, and this represents the partition between the cortex and the medulla. The upper part from this dot line is the cortex, and the lower part is the medulla. So there are three parts of the nephric tubule, that is proximal convoluted tubule, loop of Henle, and the distal convoluted tubule. Then the next part is a collecting duct. So after the fully formation of urine, it enter into each nephron, it enter into a collecting duct. This is the collecting duct, and many collecting ducts are attached to one collecting duct. Many nephrons are attached to one collecting duct, and from this collecting duct, you can see this is the collecting duct I have drawn here, and this collecting duct bring the urine into the renal pelvis, and from this renal pelvis the urine it comes to the ureter. So these were the parts of a nephron or the structure of nephron. Then come to the function of nephron. So we will start it from this Malpighian body, which comprises the glomerulus and the Bowman's capsule. So when the oxygenated blood with the waste product from the heart, it comes through the renal artery, then it enter into the afferent arteriole, and it enter into the Bowman's capsule of the, or it enter into the glomerulus of the Bowman's capsule. And here the afferent arteriole, it make a network of the capillaries. And if we see the structure of this Bowman's capsule and glomerulus, you can see here, this is the blood capillaries of the afferent arteriole, and this is the Bowman's capsule part, and this is the lumen, and there is a basement membrane present in between the afferent arteriole and the Bowman's capsule, and this basement membrane, it acts as a stainer for the ultrafiltration process. So the function of nephron, it can be divided into three parts, that is ultrafiltration, then tubular reabsorption, and tubular secretion. So first come to the ultrafiltration. When the waste when the blood it comes with the waste product into the nephron, into the glomerulus of the Bowman's capsule, here both the glomerulus and the Bowman's capsule, they act as the ultrafilters. And as you can see, look here, the afferent arteriole, it is wider than the efferent arteriole. So the blood that comes from the heart through the renal artery, it comes with a high pressure. So when it enters into the glomerulus, there will be a pressure create in the blood, and the small pores are here. You can see the blood capillaries of the glomerulus, they are formed of endothelial cells, and they have some gaps between them, and due to this high pressure of blood, they migrate from the capillaries into the lumen of the Bowman's capsule. During this ultrafiltration process, as this staining process or the filtration process, it occurs under very high pressure. This is known as ultrafiltration. And as I have taught you, this is the basement membrane. So the pores in this basement membrane are very small. So all the substance from the blood, they cannot be passed through this basement membrane. Only the small solutes, they can pass through this basement membrane. So the solutes that can pass through this membrane, they are known as the glomerular filtrate. And the rest of the residues that are the large sized large sized proteins, some corpuscles and large sized molecules, they cannot pass through this basement membrane. So they will stay here in the glomerular capillaries, and they will again pass to the efferent arteriole, and through this and the efferent arteriole, again it will make network around the nephric tube, and again it will continue as the renal vein, and again it will pass the blood to the heart. So this glomerular filtrate that produced after the ultrafiltration process, it consists of water, glucose, amino acid, urea, uric acid, creatinine, salts and some ions. And here you can see these are the podocytes. The Bowman's capsule, the inner lining of Bowman's capsule, it contains some podocyte cells. So the glomerular filtrate, they produced or they come into the lumen of the Bowman's capsule. Then it has to go to the next part, that is the proximal convoluted tubule. So after the formation of this glomerular filtrate in the Bowman's capsule, when it enters into the proximal convoluted tubule, this is known as the tubular fluid, or we can say it as the tubular fluid. And this tubular fluid, when it comes to the PCT, here the second process, that is the tubular reabsorption process occurs. And as I have we can see that the ultrafiltrate, it contains some useful substances for the body in this tubular fluid, so it has to be reabsorbed into the blood through this tubular part of the nephron. So in this proximal convoluted tubule, the sodium and water, excess amount of water will be reabsorbed from this part, and some amount of nutrient also reabsorbed into the blood. And the inner lining of this proximal convoluted tubule, it is lined with microvilli. So if I will zoom in the part of proximal convoluted tubule, you can see this is the tubular region and this is the blood capillaries that surround the tubule, and this is the basement membrane present in between the tubule and the blood capillaries, and these are the microvilli of the proximal convoluted tubule. So there is a co-transporter present in the inner lining of PCT, and in the base membrane or the basal layer of the PCT, which is in contact with the blood vessel, there is a ATP pump is present. So what is the function of these two types of proteins? And all these two types of co-transporter and pump, they are the proteins. So the co-transporter means it transport one molecule along with other one or two molecules with it. So the sodium and glucose, they come in through this co-transporter along with the amino acid, and after it come into the cells of the PCT, then through this ATP pump it goes to the cells of the blood capillaries. So after the sodium it transport to the cells into the capillaries, then there will be a lack of sodium occurs in the cells of PCT. So again, to fulfill or to replace this lack, again the co-transporter, it will transport the sodium, glucose and amino acid again from the tubular fluid into the cells, and again it will transport the sodium into the blood of the blood capillaries. Okay, so in this part the sodium, glucose and amino acid and a large amount of water, it will reabsorb into the blood capillaries or into the blood. So then come to the next part, that is the loop of Henle. And the loop of Henle, it is a U-shaped structure and it has two limbs, that is the descending limb and the ascending limb. So in this part there will be the water, it has to be reabsorbed from the descending limb, and in the ascending limb the sodium, it has to be reabsorbed into the blood. And you can see the descending limb is thicker than the ascending limb, and this descending limb, the inner lining of this descending limb, it contains some flattened epithelial cells, and this descending limb is impermeable to solutes. So as I have already talked in this part, some sodium, glucose and amino acid, they have already reabsorbed, only the water and with some salts they have retained in this tubular fluid. So it has also some, or it is also essential for our body, so it has to be reabsorbed again in this part. Okay, so as this descending limb is not permeable to any solute, so only the water reabsorption occurs from this part. So here the tubular fluid, as the water concentration it drops, so the tubular fluid it becomes more concentrated, or the solute concentration increases in the tubular fluid. Again, when the filtrate it comes to the ascending limb, here this part is impermeable to water and permeable to only the salts. So in this part, again the salt, sodium, they will reabsorb into the blood. Again in this part, the filtrate, the concentration it drops. Again, so the filtrate again it goes to the last part, that is the distal convoluted tubule, and in this part also some types of some water and sodium, they are also reabsorbed into the blood. And in this part, again in this part, the tubular fluid, it is already converted into the urine and it enter into the collecting duct. And in this collecting duct, what happens? The water and sodium again they will reabsorb into the body. If our body detect that there is lack of water in the body, so there will be secretion of ADH or antidiuretic hormone by the posterior lobe of the pituitary. Then it will make the wall of the collecting duct porous, and so the water it will again reabsorb from the collecting duct, and so with some sodium. Okay, so when the tubular fluid it reaches the collecting duct, it has already converted into urine, and after it comes to the collecting duct, then I have already talked that the collecting duct, then it pass the urine into the pelvis, and from this pelvis it will go to the ureter. So this was the tubular reabsorption, and this reabsorption occurs only to the extent that the normal concentration of the blood, it should not be disturbed. Okay, and this reabsorption occurs when there is excess amount or the loss of or any drop in water in the body, or there is, and it also depends upon the concentration of solutes it has to be excreted. Okay, so there is a third step, that is the tubular secretion. And again I have forget that this tubular reabsorption, as it only occurs to the extent of our body's need, so this is also known as selective absorption. So then comes the third part, that is the tubular secretion. In this, and the tubular secretion, it is the opposite to that of the tubular reabsorption. Here in this part, the blood or the filtrate, the blood, it contains some types of waste product like creatinine and urea, and it will again release or the secrete it into the urine that has formed in the distal convoluted tubule and collecting duct. So the tubular secretion process, it only carry out in the DCT and collecting duct, where the blood, it has already converted into the urine. Okay, so by these three process, the formation of urine, it completes, and after the formation of this urine, then it will go to the renal pelvis, and the renal pelvis, it will send it to the ureter, and the ureter, it send it to the, or it transport it to the urinary bladder, and from urinary bladder it goes to urethra for its passage. So the passage of urine is also known as micturition. So I have completed all the points under this excretion process in case of human. And so in the next class we will discuss about so we have to know about also the composition of urine. The urine, it contains 90 to 95% of water, some organic and inorganic substituents. So and it contains a pigment, that is the urochrome, and in the presence of this pigment, the color of the urine is pale yellow in color, and its pH is 6 to 8. The organic substance it contains, they are urea, uric acid, creatinine, drugs, etc. And the inorganic substance it contains are the ammonia and some mineral salts. But there is some also additional substituents present in this urine during some pathological conditions. There may be some bile salts, bile pigments, glucose, ketone bodies, etc. If we detect these substances through the urine or in urine, then we should go to for some pathological treatment. So these were all about the process of excretion in case of human, and I have completed the structure and function of nephron also. So thank you for today.